Yeah, Morris, thanks for the opportunity to talk here today. I usually talk to investors, so I don't usually talk too much about science. I talk about other things. But today I'm actually going to give you a quick summary of some of the science we're doing at Yield 10 Bioscience. We were actually founded originally out of MIT, and we became Yield 10 Bioscience about 18 months ago. And we have operations both in the US, but also here in Saskatoon, Saskatchewan. So pretty pleased with the great team we have here. We're a publicly traded company, and I'm going to show you a little bit of data, so I better tell you not to believe it because the lawyers in the US will insist on it, so sorry about that. And what do? I think, you saw in the previous talks this issue of increasing the sustainability of agriculture and the importance of yield in that equation. Ultimately, what it comes down to is Yield 10 is developing food and feed crops to produce higher yields with lower inputs of land, water, and fertilizer. We can't solve this problem just by pouring more chemicals on the fields. We've got to do a better job of the genetics, a better job of the management. We've got to manage the food waste side of the equation and all the delivery chain. But basically what we're doing is we're applying a pretty unique approach to this space, given our unique background and history. And really it's advanced metabolic engineering, and for most of you, you're going to wonder what on earth that is. And I'm not going to try to explain it too much. I'm just going to show you how powerful a technology it is and some of the things and some of the insights it's leading to. Our whole approach as an innovation company in agriculture is to solve the question of producing inherent crop yield using genetic technologies. Obviously, we're well aware of the regulatory concerns, so minimizing the number of changes and the way we deploy that technology is really important to us, so we're well aware of this. But it's a pretty small organization, about 24 people, and we've made some remarkable scientific and technology progress, which I'm going to share with you. And I'm going to speed this up a little bit because Morris has asked me to do that. So I'm going to skip a few slides, so I'm happy to talk to you about it afterwards. The whole crop equation can really be summed up as taking CO2 out of the air through plants, and plants are just a series of photochemical and chemical reactions. Ultimately, which results in plant roots, it results in stems and leaves, and the thing that we most care about for grain production is seed. And obviously, the whole mission of Yield 10 is, with the same amount of land and water and fertilizer, to actually make a much more efficient plant that just produces more seed per plant per acre, therefore having a significant improvement on essentially seed yield. So it's really about not only putting more carbon into the system, but directing that extra carbon to seed production, which is what we want to actually harvest. It's also really important to the grower because the harvested grain is really where the revenue is. Growing the biomass and the inputs at the front end is just cost. So for the farmer and the grower, revenue is the key here, and the revenue is driven by yield essentially. And then it gets really complicated. So in reality, the industry has been looking at solving crop yield and really achieving step changes in yield for a long, long time. There have been a number of technologies that have done this. Hybrid corn is a great example. Hybrid canola is another. Herbicide tolerance, BT resistance. But grain is really a sort of a computational platform that really involves three things. The basic chemical reactions going on are metabolism in the plant, gene regulation, which is pretty important, and then the environment. Ultimately, inherent yield should perform better in any growing season, regardless of the growing conditions. And that's really important given the climate change issues we face. So how do you explain this to the common layman, or somebody who's not that smart, which would be a financial guy in New York City? And the way you look at it is, a plant is really like a road system, and what's going on is cars are coming in the front end. There's a bunch of infrastructure or metabolism, and you want to direct those cars to a particular location in the system. So plants are the same. They've got all this metabolic infrastructure. This is all the enzyme reactions that catalyze the conversion of carbon. And you've also got something called gene regulators or transcription factors, and they're really like traffic lights that turn off and on various systems. So really, although it sounds pretty fancy, what we're really doing is developing a sort of a Waze or a Google Earth to map carbon flow coming into plants and directing it to seed. And in many ways, what we're trying to do is create or improve and create essentially a carbon superhighway from the atmospheric air into seed production. And then there's some really fancy synthetic biology and advanced transcriptome networking goes into this. But the real question is, does it actually work, and can it make a meaningful difference? Having said that, we are progressing a number of things because we are a business, and although we do cutting-edge science, at the end of the day, what the investor cares about is he fundamentally going to make money. And I know this because every time I meet with them, they ask me this question, and I have to justify my existence and all the expenses. Probably furthest along is the one regulated trait we would be working on, which is C3003. This is a pretty unique discovery from the University of Massachusetts. The professor who discovered that is now heading a plant science department at Michigan State University, and we still collaborate with Professor Schnell to this day. That was initially thought to be an algal gene involved in the transport of bicarbonate into the chloroplast. That's actually not what it does at all. They've learned a lot more about what it does, but our innovation model means that we have to respect our academic partners, and it's really up to them to publish and talk about the science and what they're doing. So we're not going to talk too much about that today. Monsanto's working on this. It's in their soybean pipeline. And again, our approach is get something in place with them, make it non-exclusive, let them get started on it, and let them learn whether it's useful or not in their hands. That's kind of important. C3004 is also included in that arrangement, and it's also important for seed yield. The 7, 8, 8A, B9, and there's another one, 10, are all really around oil content in cells, or in oil seeds in particular, and that's all really about genome editing. And then we have a number of the C4000 series. I'm going to tell you about one or two of those because they're pretty cool in their own right. They do apply to different types of crops, but I'm not going to go into an awful lot of detail. But ultimately, although we started working in camelina and we were working in a plant called switchgrass, which most people know is a biofuel crop, in the food sector, they're not that important. So really what we're doing is essentially canola, soybean, and corn, which in North America at least are the three big drivers of the entire seed sector, and therefore something of tremendous economic importance and value. This is what we're doing in oils, and you'll have heard some of you yesterday about genome editing and the USD APHIS decision that plants made using this technology would be non-regulated. What that does for a small company is it opens up the playing field and makes it fairer. So now we can compete, we can develop things, and there's a clear path, at least in the US. There's some indication South America is going to follow the same thing. Europe is Europe, and it's kind of unique, and we'll have to see how they decide to do this or not. Canada, I don't think, has made a decision yet, but fundamentally, it's an incredibly powerful and useful technology to really achieve in a time frame that matters what takes place naturally in the field. Somewhere in Iowa or in Saskatoon, there's probably a plant that already has these changes in it. We just have no way to identify it. And all we are doing is using a technique that allows us to identify the ones we want out of that massive population of genetic diversity. Our real focus is around something called BAD-C, which is a recent discovery, and I don't know if this works or not, but called BAD-C. This came from the University of Missouri, and it turns out to be a recently discovered key regulator of the key step in fatty acid biosynthesis. So we've actually gotten USDA APHIS approval for non-regulated status for the first one of these. We've made the triple edit. We will be applying for non-regulated status to USDA with that. And we plan to start editing the C3007. So all of this has happened in a very quick timeline and is moving pretty rapidly towards essentially development. What's interesting about this versus GMO is once you've made the edit, you have your product. You don't have a development platform, you have a product. And so although we weren't really doing this other than as an example of using a platform to develop the technology in-house, it is something that can be progressed commercially if, in fact, there's interest in doing so. Now, having said that, I'm going to flip now over to the second area, which is this is really about traffic lights. And what we were looking for, to use an analogy, was global transcription factors, or a master switch that controlled all the lights in the city and allowed us to switch on the ones we wanted and turn off the ones we didn't. And we had no idea what these were, so we developed a unique approach to mine public data and identified three really interesting genes, and we smartly named them 4001 to 4003, just to imply that we'd done a lot more work than we actually had done. But we have a great group of people, so they're just really, really smart, which helps a lot. And what we did with these is we actually, we engineered them in switchgrass. We just introduced them in there and we made them much more active. We turned them on. So we turned those lights on so that they're now on full green. And they have a pretty dramatic impact in these plants. All three of them had similar impacts, probably for different reasons. The most interesting thing about them was the amount of biomass produced went up by up to 160%. So that's almost 2.6 times as much plant as the non-traditional plant. This is a switchgrass gene used in switchgrass. So if one wanted to, one could do this in other plants using the native gene. And of course, GIL10 has identified the genes in corn, soybean, canola, wheat, rice, et cetera, And as a small company, we're very busy patenting all of those because that's really important to us. The other interesting thing is the below-ground biomass in these plants increased enormously. And there is a concern emerging in the Midwest, particularly around organic soil content. And that's really important for the long-term sustainability of agriculture. So that's pretty exciting stuff. We would probably never commercialize this this way. But what's really unique about it is now that we know what success looks like, we can actually study it. And we can then begin to reverse engineer and look for alternative ways to get somewhere towards that performance with perhaps less dramatic changes. This is just another example. Really, basically what happened at the very front end of these plants is photosynthesis went up enormously by about 70% as measured by electron transfer rates in this case. So they're just much more efficient. We didn't have to apply more fertilizer or water or anything else to these. In fact, the plants are also drought tolerant. And it turns out there's a good reason for that. They switched on all the stress tolerance genes as well. So this is a pretty cool tool, actually. Pretty exciting results. It was recently published, so anybody who's interested can get more information on that. What I think was more important for us was looking at the genes or the transcription factors that had been turned down. In other words, we'd switched these lights off, if you like, and they were no longer blocking the flow of carbon in the plant. And there was only 5 downregulated. And when we looked at the same sort of data from the other 2 global factors, it turns out there's only 22 transcription factors that are down-regulated in these plants. It's not a large number, given that there's 30-odd thousand genes in a plant like corn, 2,600 transcription factors. So we sort of narrowed that down to 22 targets to essentially turn down using genome editing. There's only one of those down-regulated by all 3, and it turns out to be an incredibly powerful regulator of plant growth. So if you make more of it using gene technology, you get 2 inches tall switchgrass plants. We've obviously more interested in knocking that out, and we've actually done that, but we've done it in rice for a whole set of other reasons. Again, that under USDA rules would be essentially a non-GMO crop with hopefully a better yield potential than that. So this is something that's pretty unique to GIL10. I'm absolutely not going to try and explain this. A, because I'm not capable of doing that. But B, really, it allows us to model essentially carbon coming into a plant, to track every reaction in the plant, and to determine where that carbon is going, and all of the things that have to happen to essentially enable that to be most efficient in directing carbon to seed. And so what's cool about it is you can reduce botany on the left to essentially something that looks like a circuit board. And this is a kinetic model of the mitochondrial, which is like the energy or the battery sensor in a plant. I'm not going to tell you what's been done to that plant on the right-hand side, but obviously it looks kind of more interesting than the one on the left. That's a story, obviously, for another day. This gets more complicated from sort of the engineering point of view. And this is kind of what a flux model looks like. And to most plant scientists or biologists or people involved in gene expression, they go, what the heck is this? And what does it do for me? And how do I use this? So I'm going to show you just basically how this works. And this is just an example. I've had to delete some data from this because it had some stuff in it that's a little pre-competitive, I guess. And this is a pretty competitive field, so we have to be aware of that. And all you have to look at the bottom is, what we did is we built these models for essentially boosting seed yield, looking at photosynthesis in green tissue or leaf tissue. And keep in mind in oil seeds, there's a lot of photosynthesis going around, the side leaks and the seed pods and what have you as well. And there's only really one case that's really interesting, and that was the potential impact of one of these synthetic biology carbon-fixing pathways in seed yield, assuming one could actually do that. You could potentially, without adding anything else to the plant, you could just make it double seed yield, essentially, just based on carbon conversion efficiency, nothing else. You don't need anything else. It's just a much more efficient plant at taking the CO2 it's already paid to fix with the nutrients it's already absorbed, and it just makes more product for the same input, which ultimately, that's the key to sustainability. Big question is, does it really work? And so I'm going to show you. Because modeling is a cool thing, but quite frankly, you can really fool yourself and go down enormous numbers of unuseful tracks through modeling. And it's only useful if you can really challenge those models with experimental data. So because we're scientists and because we came out of MIT, we did the experiment. And actually, for the most part, we did it right here in Saskatoon. So this is a complex carbon-fixing pathway. Plants don't have this, so we introduced it into the plants. There's 12 genes on here, so obviously not something you'd commercialize. But there is a really important question, which when you think about global food security, and just taking into account all the different approaches, there is that fundamental question, could you actually do that technically? Could you make a plant make twice as much seed? And that's just an interesting question to ask for a company like Yield 10. So we made, essentially, typical vectors to do this, and we introduced them into Camelina sativa, which is a plant we've done a lot of work on because it's very fast, it's very amenable to genetic technology. And we made a whole bunch of these things, and we looked at the best ones. And interestingly enough, the time we were doing this, really the focus of the project was on oil content for a biofuel project. And the scientists got excited about the increased oil. But when you look at the impact on the seed yield, it's a little bit more interesting. So if you look at the best lines, and I think they're sort of highlighted at the bottom there, and these two yellow things, essentially the seed yield's gone up 2x. The plant's essentially the same, but you're just making twice as much product, which is a pretty interesting result. And it's happening in a number of different lines, so it's real. The other thing that's happening is the seed size or the seed weight has actually increased by 30%. So it's a combination of more seed, but also bigger seed. And there is certainly some evidence that the oil content in those seeds has increased as well. So this is probably a first in terms of proving that you can actually use this type of advanced technology to basically double seed yield in the plant. Obviously, we have to be realistic, however, in terms of how we think about this. It's a tremendously valuable tool. It proves the point you can do this, but we don't really see this as something that we'd ever consider commercializing. Now, if everything gets really bad in 30 years' time and we're starving, I'll change my mind on that. So in terms of the conclusions from this, doubling seed yield, at least in Camelina, is certainly achievable. We've actually made a lot of progress on canola and soybean as well. I'm not going to talk about that. Carbon conversion efficiency is really a tremendous way to boost yield with the same level of inputs. That's sort of an interesting outcome from this. And these new types of modeling and these new types of synthetic biology technologies can be applied here. It's really not suitable for commercial deployment at this stage, and we wouldn't be planning to do that. But what it has done, it's allowed us to analyze those plants, to analyze other changes, and identify a series of plant genes that can be modified through genome editing to accomplish similar things. So even if we don't get to the 200% or whatever it was, ultimately what matters is that we can boost yield in these plants by somewhere around 20%. And that will essentially meet all of Yield 10's technology goals. And to do that in a way that's readily approved through the regulatory process and broadly acceptable to the public as a whole. I'm going to skip this because it just kind of explains in more detail what this grain platform is about. It's actually pretty cool technology and involves some really cutting-edge science, both in the chemical engineering side but also in the plant science side. And so in summary, it's a pretty unique approach. It's not something the industry has really been involved in to any extent. You're beginning to see it appear now. But Yield 10 is this very unique 30-year track record of success in this space. And so we have some very unique knowledge and background and history that allows us to do this super efficiently. For us, as a small company, We like to partner with people. We're actually great people to work with. We're fun to work with. We have a good time. We work hard, but we enjoy ourselves. And hopefully we're doing something that can really contribute to humanity. Separately from that work, obviously the oil traits are progressing pretty aggressively. They would be non-regulated in the US, and we're moving those also into other commodity crops. We do employ both GMO and editing approaches to achieve our goals. We are only too well aware of the GMO question, but we have to also, as scientists, be honest with ourselves, and the scientific data basically speaks to the safety, efficacy, and the sustainability benefits of this type of technology. And really, ultimately for Yotan, it's all about licensing and partnering and working with other people throughout the ag sector. So we do partner, we're open to working pretty much with anybody, and we'll figure out a way to do it if it makes sense for both parties. So with that, I'd just like to re-thank Maurice for the invite to talk to you about something that's completely off the wall a little bit, but really kind of exciting if you really focus on this as a technology and something that can make a meaningful contribution to the overall success of achieving yield, basically food security. So thank you, and thank you for listening.